In the previous class, we have discussed about introduction and historical background of biotechnology. Today, we will deal with the topic of principles of biotechnology. Principle. Principle includes two techniques. First, it is genetic engineering. And second, it includes the second technique, which is maintenance of sterile ambience. Genetic engineering. Engineering, as we know, it is a technique to breaking and joining. What it include? Breaking and joining to create new combinations. And when this engineering is done at the level of genetic level, at the level of genes, then it is termed as genetic engineering. So what is the outcome of this genetic engineering is? It is just alteration of chemistry of genetic material. Chemistry of genetic material. Then this altered and creating the new combination. After creating this new combination of genetic material, this is introduced into the host organism. The second step is also very important. Introduced in host organism, where this altered genetic material will multiply, and after its multiplication or due to multiplication, the phenotype of host organism will change. So we'll get the phenotype which is desired. This can be in form of any molecule or any biological function. So phenotype of host organism we got change. Why we are getting this? As we know, again I am just saying, repeating it for again and again, DNA will form RNA, it will form protein. This protein will change the biological function. The same is going on here. So the phenotype is going to be affected by this new combination of genetic material. Then the second, what I have said, the maintenance of sterile ambience. When this altered genetic material with a new combination is introduced into the host organism, this host organism will be cultured in the lab, or in vitro culture will be there. At the time of its culture or at the time of multiplication of host organism, the very important thing is that the whole system should be free of any contamination. So its maintenance, okay, what is that? Free from contamination, so that we will get only our desired product alone. Okay, the multiplication of host organism, it can be either any microbe or any eukaryotic cell or whatever. Multiplication of host organism should be alone. so that we get desired biotechnological or biotechnology based product. When we combine these two, we get the biotechnology principle. So it was the whole about principle of biotechnology. Now let us see how did it extend. So just let us see once again. Principle, it includes the two techniques, genetic engineering and maintenance of sterile ambience. In the genetic engineering, we just change the chemistry of genetic material which create a new combination. This new combination of genetic material will be introduced into the host organism. This host organism will multiply and we get the desired phenotype or desired protein or any biological function. At the time of multiplication of host organism, it is very important that that system should be sterile, means it should be free from any contamination. When it will be free from any contamination, the multiplication of host organism will be allowed and we will get only our desired biological product. This is the main principle of biotechnology. Now let us see the first formation of recombinant DNA. Before discussing it, there is a concept of ORI. It is very important. ORI. What is this? Origin of replication. Origin of replication. In any chromosome, it is a very important or a specific point which is responsible to initiate replication. It is responsible to initiate replication. We will discuss it later in detail, but here I am just introducing this keyword because it is very important. And when we insert our desired or required DNA piece into the plasmid and creating, suppose it is our desired genetic material and it is that DNA piece where we are just creating the new combination as or the recombinant DNA, and then it will introduce into the host organism where it will multiply autonomously and we'll get the product. Multiplication will there, and we'll get product which is required. The multiplication will be only done when it will go for replication, and this will go for replication only if our desired DNA piece is joined at the ORI, origin of replication. Otherwise, its introduction or creation of recombinant DNA will be failed because its multiplication will not happen. So, it was about ORI, which is responsible for initiating replication. As we have discussed about principle of genetic biotechnology, then there was an important keyword which was ORI, origin of replication. I had only introduced that topic, then that term, and then there is an important thing, how the first recombinant DNA has formed. It was developed by, again very important, Herbert Boyer and Cohen, Stanley Cohen, in 1972. How they have done this? First, they had isolated. First, we can say they identified the desired DNA, which was antibiotic resistance DNA. It was antibiotic resistant DNA. Okay. Then they isolated with the help of restriction enzyme. As I have told it earlier, restriction enzyme is that enzyme which cut a DNA into pieces at a specific site. So this restriction enzyme is also known as molecular scissor. They isolated this desired piece of DNA and then they linked it into the plasmid. Again, I am saying, what is a plasmid? It is the extra chromosomal part of prokaryotic cell and which multiply autonomously or independently. Plasmid of Salmonella typhimurium. And they get they get this recombinant DNA. How they have done? As I have told, they identified the piece of DNA, then they isolated with the help of DNA, with the help of restriction enzyme, then they joined it with the plasmid of Salmonella typhimurium. With the how this joining is done, let us see with the help of some diagram. If it is your desired piece of DNA and it is your plasmid, then it will be as recombinant, and this recombinant plasmid, when we are going to join this is done with the help of an enzyme which is known as ligase enzyme. So there are two most important enzyme in biotechnology are, first it is restriction enzyme and second it is ligase enzyme. With the help of ligase, it is used as a ligating, joining two pieces of DNA at a specific site. Then after when we are just joining this two different pieces of DNA, we are getting this recombinant DNA. Then this recombinant DNA is introduced. This recombinant DNA introduced into the next host organism, which was E. coli. Introduced in host organism. That host organism was E. coli. In the E. coli, it started to multiply autonomously, and we get our desired product. And there will be multiplication. Now, how this multiplication is going to occur? This piece of DNA or recombinant DNA will use the next third enzyme, which is also very important. That enzyme is polymerase enzyme. Polymerase enzyme of host organism. Okay, so it is our third enzyme which is again going to play a very important role in biotechnology. With the help of this polymerase enzyme, this plasmid will get multiple copies. And after getting its multiple copies, just like this, as the cell of E. coli, suppose it is cell of E. coli, it is its main DNA part, and it is its plasmid or recombinant DNA. As this E. coli will multiply, the same number of plasmid will also multiply, and after its multiplication we will get a desired product. So just let us see once again how the first recombinant of DNA was developed. With the help of or by the contribution of Herbert Boyer and Stanley Cohen in 1972, they had just isolated a desired piece of DNA that was an antibiotic resistance DNA from the Salmonella typhimurium, and they just isolated DNA and they formed the recombinant DNA by joining it into the plasmid of Salmonella. It was something like isolated piece of DNA. Then the plasmid they inserted in isolation. Then the second introduction in host organism. And then last step is its multiplication. So it was all about its principle of biotechnology. We have discussed its introduction, historical background and principle. In the next class we will discuss the tools of biotechnology, which is very important, and so I hope you will continue it for the tools of biotechnology. So it was all about the principle of biotechnology, and thank you so much.